started, I just want to thank everybody for attending. This is Labshire's Ask the Expert series. This webinar is going to be inside the FDA's proposed rule to regulate laboratory developed tests. My name is Jenny Bardsley, and I'll be serving as the moderator for today's webinar. During our time together, you will learn or understand the implications of the upcoming new regulations, learn how to approach a risk assessment and remediation plan, and discover how to address gaps efficiently and effectively. After our presentation, we will have a Q&A. I see that some people have already submitted questions during the registration process, so I appreciate that, and we'll address those as well. You can submit your questions by using the Q&A button, and there's also a chat button, and we will answer those questions, like I said, at the end of the presentation. However, now I'd like to introduce you to one of our panelists, which is Dr. James Blackwell. Dr. Blackwell has been a prominent industry consultant for over 17 years, working on a wide variety of CMC and quality issues for a broad range of companies, investors, and product types to all stages of the product life cycle. Prior to consulting, he had several senior positions in technical operations, manufacturing, and process development at prominent operating companies, and has been on a founding team of several startups. Now it's my pleasure to hand over the presentation to Dr. Blackwell.
Next slide.
Thanks, Jenny. We welcome all our attendees. We think we'll find great benefit from listening to our experts and our thoughts on this. This is a very important issue that is going to happen. It's just a matter of timing and when. And as you look through the slides here, I think there's really several themes here. It really is important to implement these kinds of best practices. We've seen on the forum discussions and things a lot of angst on people, on the part of people who may be impacted by these new regulations. And I get it. I think earlier in my career, I probably would have been more sympathetic. But as I've learned more about quality, and quality has become an important part of our practice, and we've worked closely with clients implementing quality systems, what I've learned is that if you implement an efficient, well-designed quality system, it really makes your operations more efficient, more compliant, and it's really best practices. So one of our themes today is really that implementing these things sooner, doing it in a risk-based approach, so it's designed to focus resources on where they're most needed for your particular application, will provide business benefits beyond just being compliant. So with that, I'd like to introduce our panelists, Mark and Deborah, who will do self-introductions.
Hi, thank you for joining us. I'm Deborah Miller, and I have in excess of 40 years experience in the laboratory field. I've worked with clinical labs, research laboratories, pharmaceutical, medical device, forensic toxicology, and have been on the operations side as director of a lab, and I've been on the quality system side. Some of the things that I consult with are regulatory and industry standards and requirements, remediation. I've worked with 3 consent decree companies to help remediation. We've done some laboratory design literally from the concrete up. I've been a CLIA and ISO 15189 inspector, and I've trained on folks on ISO 15189 and have provided many training classes in the regulatory field.
Good morning. I'm Mark Gusak, another principal consultant. I started out in this field as a nuclear medicine technologist, then graduated to a clinical engineer. And I've been a licensed healthcare risk manager in the past, but in a broad sweep of my last 40 years, I've been a board certified pathologist, both AP and CP. And my experiences cover really all areas of the clinical laboratory. I've held jobs ranging from a bench technologist as a nuclear medicine technologist, all the way up to directorships of small hospital laboratories, large reference laboratories, and even hospitals themselves. So I've kind of had both a technical and a broad strategic view of the laboratory field. And I too have been involved with laboratory design, construction, instrument procurement, and validation, as well as moving laboratories. I've been involved with laboratory accreditation to clear regulations, as well as ISO 15189 standards. And I focus a lot on systems analysis, process control, and document management. That's kind of the core of a lot of what I do. I've been involved as well with quality systems design and risk assessment. And then I have a background in information technology, which I apply to the laboratory field. So we're going to begin the presentation now. I'll be the primary speaker, but occasionally you'll hear Deborah pipe in. She's not interrupting me. This is a plan. That's how we deal with this. And really what we're talking about is the FDA proposal to reclassify laboratory design tests. And that would require, meeting a much stricter and more expensive regulatory oversight. So our question to you is, do you have a risk assessment plan to meet this challenge, regardless of what happens with the proposals that the FDA has brought forward? So we're going to start with history. What I like to do is go over history so we all kind of have a same reference frame, then look at what the situation is, then look at what the impact is going to be, and then finally, how do you assess this impact and do things? So this has to do with the FDA Code of Federal Regulations 29 CFR 809. And that was passed by Congress almost 50 years ago. And its primary purpose had to do with in vitro diagnostic products. So it was designed to provide a means for regulation of commercially developed laboratory test systems, not those that were carried out by individual laboratories for themselves. So as we all know that IVDs are designed to detect and measure biologic constituents of human blood, body fluids, and tissues. And for that reason, there's an issue. The regulation purpose was to assure a minimum degree of safety, that is, so that the patient would not be unduly harmed, either by overdiagnosis or underdiagnosis in the face of the type of therapy that might be available, the quality of their experience, and the utility of it in terms of the cost versus the what they get as a benefit. So at that time, many clinical laboratories served a limited number of clientele, and they were exempted from this the 29 CFR 809 by the FDA. And the reason for this is because they needed to design in-house tests that were not readily available from commercial sources. There are tests that the companies that are for-profit companies simply couldn't see investing in, especially because they had to put all of their instruments and test methodologies through the FDA, and that's a very expensive process. And at the time, these were considered to be of low risk to the patient. And so really the FDA felt that there didn't need to be a formal approval.
And Mark, I'd just like to jump in here and remind everybody that this was before CLIA 88 came out. So
Right.
just as far as the FDA's approach.
Deborah, could you explain what CLIA 88 was?
Well, that was the regulation for laboratory medicine that CMS, that is the government agency that implements it. And that's the regulation that the accreditation of clinical labs is based on. And if you're operating a clinical lab in the United States, then you have to be, depending on the complexity of what the testing you're doing, you have to be certified by CMS as a CLIA accredited laboratory.
Thank you.
Mm-hmm.
Yeah. And so the FDA classified these tests as laboratory developed tests, or LDTs. So more history, how things change. Over the last 50 years, it's been an incredible advancement. The complexity of these tests, scope of application, range of clients using LDTs, it's all expanded greatly. And that has raised concern over safety of these tests since they're not held to the same standards as IVDs. And the laboratories that develop these in usually large hospital complex systems, the concept of quality versus the utility value has not really been addressed. So as a result, the FDA became increasingly concerned that some of these LDTs might not provide reliable and safe results. And that's the issue here, is patient safety. That comes first. So in the past, the FDA has attempted to implement rules to bring more stringent oversight to LDTs, but they've all been unsuccessful. But more recently, the FDA proposed really a sweeping new rule, as we'll see in a moment, to bring these standards up in line with IVDs. So the situation, here's what we're going to talk about now, is the recent proposal itself. And that was in October of 2023 when they published the proposal to change how the law is used to cover these tests. And this involved the addition of the following 10-word statement that redefines the meaning of manufacturer. And basically what they're saying is that now the laboratory is considered a manufacturer of these products. And therefore, if you look at the entire regulation, they had separated manufacturers from the laboratory. So they could say laboratory developed test versus manufacturer developed test. Well, now they're all the same. That brings them under the cover of the IVD. And that means that there are a lot of additional work and costs that laboratories will face if they have to do this. And the proposal is that it will be phased in over a 4-year period. And for us, that sounds like a pretty reasonable amount of time. But if you talk with people in the commercial side, the vendors who sell and the manufacturers who make, 4 years is a very short period of time to get everything done for FDA approval. The other thing is the amendment does not include a provision for grandfathering in LDTs. So if you go back 40 years, what you find is immunohistochemical stains on tissues have been grandfathered. Now, there are instruments that will automate, and those are FDA approved, but the actual use of them and also modifications of the manufacturer's reagents or the way the instruments are used is not covered. That's been grandfathered. So in response, there's been a significant groundswell of the healthcare industry to prevent this change in the regulations. And the pushback, we realize that this could lead to disapproval or alteration of the amendment, or delay in the phase in period, or litigation, or they might even create a list of characteristics or criteria as to what they will redefine. However, we also know that when you're dealing with government regulatory agencies, you can't really predict the outcome. So now we look at the situation. Oh, you go ahead.
And speaking of government regulations and not being able to predict the outcome, I would suggest, next slide, don't rely on the potential delay. Look at your current process to ensure that your clear regulation, 42 CFR 493, that go ahead and advance it, Mark, and that particular standard is met now. Even though you're a CLIA accredited lab, as a result of this LDT deliberation, there may be renewed interest in your current qualification validation records. So you've been inspected, somebody has looked at your laboratory developed test records, and you've been reaccredited. It's possible that given this renewed interest from all sides, that your next surveillance assessment may look at these records with a bit of a different standard. Like much of the CFR, it tells us what has to be done, but not how to get there. So the idea of what the standard is very general. Well, we know that currently it includes supportable data for imprecision, accuracy, interfering substances, matrix effects, clinical reference ranges, traceable standards, controls, and proficiency testing. That's the minimum required by 42 CFR 493 now. As part of your risk assessment, it would be good to revisit any of your LDTs that are currently in use to make sure that your data is documented and holds up to the scrutiny. And adequate degree of test verification is a broad field that can a lot can apply to that, and it's up to the assessors to make that determination.
So we've seen the history, and also the fact that it's not just the FDA, but CMS and other agencies that can intervene, and they're aware of this, what's going on. So we have to look at the potential impact on the laboratory. This is just one slide. There are many other impacts that could arise as this moves forward. So the things that we need to understand, even if the amendment fails, are the following. So the public area of safety concerns combined with the rise of LDTs being made available for a fee. A lot of these LDTs are related to the field of oncology and precision medicine, and these drugs that manage tumors through their physiologic and biochemical pathways. And so, in order for them to do the clinical trials and also to determine which patients should get these drugs, a lot of LDTs have been developed. And they're being used by not just the hospitals where they're being developed, they're being used elsewhere, and they're being made available for a fee, which is a big fee. So whether or not the FDA succeeds, there's a real potential for increased liability, because this is now out there, and you know that there are going to be other people looking at this. So there's the issue for facilities and physicians that use LDTs, both for these clinical trials as well as the patient care. Next, there's the liability of those sending specimens out to other laboratories that use LDTs to generate results. And then finally, pharmaceutical companies submitting trial results to the FDA for approval may also be impacted if they're using LDTs, and I can tell you right now that they are. So regardless, continuing use of these LDTs or rolling out new ones may increase both liability costs for the laboratory, as well, of course, just the cost of getting it through the FDA. And so you're going to need a lot of resources to meet this challenge. And so you need to, begin to get ready. What we can do, of course, is we can take the risk approach, prepare for the worst, and hope for the best. So there are numerous pre-market reviews to assess the reliability of IVDs, and therefore it would also apply to LTDs regarding their specified use. And that means that if a laboratory has an LDT and they're putting it through the FDA, and there's clinical trials going on, or there's some change somewhere in the criteria being used, or how they use it, they would have to start all over again. They can't just simply continue to modify the LTD while they're going through the FDA. The more stringent standards for proof that the test can achieve a minimum degree of accuracy and precision will be involved. Then there's the risk of an expensive development where you put it all together, and then the FDA says, no, we're not letting you through at this point. And then, of course, if you've already deployed them and it doesn't make it through the FDA approval process, that's a real critical issue. And of course, we have no idea whether they'll include a grandfather clause in the future, or whether they will have criteria as to which ones they're going to go after. Obviously, they can't go after all LDTs at the same time, but we can be assured that somebody is going to be facing this if this gets approval. And then there's the risk for increased malpractice lawsuits being brought against those providing or using LDTs. Either the LDT leads to a very dangerous therapy that's more dangerous than really what the patient should be getting, or the patient is denied an effective medication because the LDT said that they shouldn't get it when in fact they should.
Or the patient could have an adverse outcome that has nothing to do with the LDT. However, because they used an LDT, that gives an opening for litigation because of the controversy around it.
And that has to do with the basic premise of risk management for those of us who've really done it professionally. A lot of people look at frequency and rank severity. It's perception. In the end, it's all about perception. And so a lot of this is that's now in the literature with people arguing back and forth is going to heighten that whole perception issue. So now we move on to the assessment. So given the number of high complexity LDTs, which are presently being used, combined with the rate of growth in this number, and given these tests are often offered for a fee to other facilities, at least some will eventually be reclassified as IVDs. I just think maybe if not now, in the near future, because I've dealt with these, and a lot of them really need to come under higher scrutiny. So this will occur through one or more of the following. So FDA actions that bring some or all the LDTs into the formal approval process. Congressional legislation, perhaps to update 21 CFR 809 to include LDTs that are marketed or that meet a complexity measure. So because of the fact that there will be lobbying, I'm sure this will get up to the congressional level. You may see that it occurs at this level, not at the agency level. Then there could be targeted regulatory action or litigation against facilities marketing these LDTs, whereas those that are using them locally will be left alone at that point. So what we're saying now is that organizations, they're going to have to be, if they're looking into implementing them in the future, they should consider planning prospectively to meet these new regulations. And that's where you plan for the worst so that you're ready and you know what's coming, and you're not going to be caught, with your laboratory pants down, instead of just simply walking into a crisis. If the organizations are already using them and offering them, especially outside their facility, they should consider retroactively meeting new requirements and figure out what that's going to involve. And then assessing the current situation regarding LTDs in the facility, and that all assessments and decisions are documented. So you can show, we realize this was happening, this is what we've done, this is how we've planned. Because you can never tell, you can have a change above you in the administration, a change in the laboratory, and a change in who's inspecting you. So what we're saying is, do not wait until it's too late. You should really begin to start a formal process now. So our recommendations has to do with this readiness that we're talking about. So you should determine if you have LDTs, or use those sourced from another laboratory. And if so, perform a comprehensive review and then upgrade the laboratory operations. And we advise the following, which include but are not limited to a really strategic level risk-based crosswalk and gap analysis. You want to find out, you may be doing LDTs, you don't even realize it, that's what they are, because they've been going on for 20 years, since before you got there. Or you're sending stuff out and you really don't realize that reference lab, actually what they're doing is a modified test and it's an LDT. And you want to find that out.
Remember that the regulation includes modified FDA approved tests. So it's
Right.
important to know that a lot of labs could modify the volume of the sample for pediatric patients, for instance. If that's not in
and also for for dialysis laboratories, they do that. But also, I in doing ISO 5189 appraisals, especially molecular biology, in flow cytometry, cytogenetics, I see a lot of that. And what I see looks good, but if the FDA gets a hold of those, they're going to have to completely reevaluate this whole situation. And of course, as Deborah has talked about, the current 42 CFR 493 requirements, because that can become an issue, because the people who do the inspection may be required to look more closely at LDTs. And you want it to be a science-based risk assessment. You look at each gap identified and determine the degree of remediation that's needed. Because there's some where you can upgrade them and it's going to cost you a whole lot of money to be ready to be either inspected or to have to submit that. So you then develop as cost effective as possible remediation plan, and you focus on the safety and the quality and utility aspects for patient care. As long as you're focused on that, it's very difficult to go wrong. Yes, there are all sorts of technical issues in filling out all the forms for the FDA if it gets to that, but those are things that you can find on the internet, or you can bring in people who are expert on that. So you implement the risk-based management system to cope with ongoing compliance issues, because this might just be the first of many volleys that will come across our laboratories. And then any delays in the final amendments do not negate the need for assuring safety, quality, and utility for the patient. I mean, one of the reasons why we have regulations is because over the years there's been a lack of attention on this, leading to high levels of litigation, and then legislation follows suit, and then regulation follows suit, and then we find ourselves in a bind. So institutions without the knowledge to implement change to meet the new requirements should consider bringing in one or both of the following: additional personnel with the appropriate qualifications to do this. I can tell you that's very hard to do. It's also very expensive, and now you've got another full-time employee. So also consider consultants with expertise in regulatory requirements and quality systems to get the help to get the ball rolling. So you do all this, and then that means that you can successfully meet the challenges posed by changing landscape of regulatory oversight, not only of LDTs, but the laboratory in general. So the conclusion is facilities with clinical laboratories, the laboratories themselves and clients using LTDs should do the following: closely monitor upcoming changes. Okay, you want to keep track of what's going on. You want to do your risk assessment of your laboratory operations. And since you're going to be doing this, you might as well look at the entire laboratory from a strategic point of view. And that's why we talk about looking at this strategically, not just simply focusing in on one little thing. And think about how they could be impacted by changes. And then you follow that with a plan to remediate any issues identified that may require compliance with FDA rules. And then you seek the expertise needed to succeed in responding to present and future situations. So we ask the question again, do you have a risk assessment plan to meet this challenge? And if not, the people talking to you can help you. So before we get on to the questions, I just want to briefly say that the final rule is coming in April. And regardless of that, we plan to do in our next presentation a look at all the phases that are proposed by the FDA and how one should plan for them to understand the implications of coming new regulations, learn how to approach a risk assessment and remediation plan, and then discover how to address gaps efficiently and effectively. So now on to questions. We'll let Jenny take over and provide those.
Sure. Excuse me. Thank you, Mark and Deborah. Like I said, we're transitioning over to the Q&A portion, so continue to submit your questions on the control panel. And I'll start with our first one here. Let me see. In regards to impact on CMC and clinical testing, what phase of clinical development is this meant to cover?
Well, that's a really good question. So let me clarify. Are you saying which FDA regulations would be required if LDTs are required to meet IVD standards?
Mark, I think the question is for a clinical study. The word clinical in that question, I think they're talking about clinical studies. So we talked about if a clinical study is using a LDT to manage their patients in the study. So the question is what phase, I guess, of the development of the drug are we in?
Okay.
So James,
I thought we were talking about the LDT. Yeah, I can't answer that. So here's the problem. The initial phase is just to determine, what the risks are. I mean, is this a drug that does so much harm that any possible benefit is canceled out, and so it wouldn't make it through. The phase 1 trials wouldn't make it through the FDA. So the issue here is if you're using an LDT right at the beginning, that in and of itself is going to be a problem. And I think that the FDA would probably require the LDTs, even at the phase 1 trials, to be brought through their process. And then, of course, phase 2 and phase 3, are going to be dealing with large numbers of patients, and of course, that would be considered necessary. So I think you'd be stuck all 3 of the phases.
And so,
yeah, I agree with you. I think the question was directed towards phase of clinical development, phase 1, phase 2, phase 3. And often what I advise clients, if you don't have a regulatory roadmap for something, look at other regulatory guidance and look at the rationale. And I think to tee off Mark a little bit here, I think a risk-based approach would focus on those assays and methods and tests that are critical to patient safety. In which case, there are other examples, right? And we hear about phase appropriate GMPs and those kinds of things. So in this case, if a particular test was critical to the patient's safety, then I think starting at phase 1, then it should be a client. It should be an appropriately validated and assessed LDT for that purpose. And then I think those standards would increase as you go through phase 3 towards commercial.
And I've worked with companies that are setting up clinical trials with a partner hospital, and their user requirements are typically a CLIA accredited lab. They don't typically look at the assay in any more scrutiny than that, because their focus is, if it's CLIA accredited, it must be okay for our study. But now, when we see this murkiness around LDTs, there may be more that needs to be done to establish that relationship with that laboratory.
Okay, thank you. We'll move on to the next question. And this one is, how long would it take to get ready if you haven't practiced these things to date? 3 months?
Well,
That depends on
I'll
the test
start.
itself.
I think if we're talking about a small test, in terms of coming in, doing the gap assessment, risk assessment, and implementing a remediation plan, I think that's certainly possible within 3 months. And depending on what the outcome of that and what the remediation plan is, it could take longer. But I think in terms of getting the baseline and getting started, that certainly could happen with 3 months. But I'm open to hear what Deborah and Mark have to say on that.
Yeah, I think it depends on the high complexity, medium complexity of the assay, and whether or not it's a manual or a automated instrument, whether or not it's a modified test or a start from scratch laboratory developed test, all of those things. The assessment, I agree, James, could take 3 months. But then again, as you said, the remediation would depend on what the assessment determined.
Yeah, I have nothing else to add to that. I
Okay.
think that we've covered that question. That's excellent.
All right, thank you. The next question is, did the FDA establish criteria for LDTs back when regulations were published?
Do you want to handle that, Deborah, or do you want me to?
Well, I would say not for LDTs, for IVDs. They did not make the connection with the IVD requirements for clinical laboratory developed tests, Mark.
Yeah. They basically grandfathered a lot of things in. All immunohistochemical stains are basically LDTs, although there are automated instruments that will run them. The actual technology that was developed, the basic methodology, is an LDT. It still is, and it's still being developed and used in a lot of oncological fields.
Okay, thank you. Another question is, what does CLIA mean by adequate test verification?
That's the $100 question, $1,000 question.
I think it's $1 million question.
Yeah. That's the reason that I brought that up, because that they tell you what has to be done. They don't tell you how to get there. And you just have to make sure that your statistics and your data and your number of tests and your population that you determined your reference ranges with all meet the typical requirements. Where can you find these requirements? You can find these requirements on what they put the requirements on the IVD manufacturers as a start. Mark, you want to talk a little bit more about the data that supports a validated test?
Yeah, basically what we're talking about is the field of metrology, or measurement uncertainty. And the value of a test is going to be dependent on the accuracy and precision. Basically, it's the reliability of the test. And the less accurate and less precise, the higher the risk is for an inappropriate outcome, where the patient has a disease or a certain physiologic state, and the test does not confirm that. It just confirms that it's a discordant result, or vice versa. You get an overcall, and then the patient either doesn't get the right therapy or gets the wrong therapy or dangerous therapy. And this all comes down to, well, what are the benefits and risks of that test? And obviously, if it's a high-risk test, you need a much higher degree of accuracy and precision. And that's an open-ended question as to what is adequate. But there are some rules of thumb. We're not going to get into them now, but in terms of the number of samples you would need, how you would stratify those, and what kind of statistical analysis you would carry out to determine that what you've got is adequate to protect the patients as much as possible.
And just the simple things like your standards and your controls. What's your source of your standards and your controls? Are your standards traceable to a NIST measurement? Do you know the matrix effects? Do you know interfering substances? That's all in the regulation now. So to answer the question, do you know, you have to have the supporting data that you've done the work with the unknown, with the test, to make sure that you can prove, yes, we know.
Thank you for your response. This person is saying, you said laboratories that send specimens to other labs reporting out results using LDTs are still responsible for those LDTs. Why is that?
Yes. Interestingly, the pharma and medical device industry is very used to supplier approval for materials, for their raw materials for their manufacturing. Clinical labs probably haven't been exposed to that much, but when you pick a reference lab, most labs probably think, is it CLIA accredited? And if it's CLIA accredited, then you're confident that you're getting results that are reportable to your clinical staff. However, now understanding that the CLIA accreditation doesn't currently really vet the LDTs that may be happening in that lab, your lab, since you are the one that is picking the lab and ensuring the clinician that you're getting a good result, has a higher degree of responsibility to understand the reference lab that you're sending it to. It could be a questionnaire that you go into more detail about whether they have LDTs and how they've qualified them and how old they are. It could require an on-site assessment, a supplier assessment on your part. But since you're the lab that the clinician has sent the specimen to, you've chosen the reference lab, and you're reporting that back out to the clinician. That's your responsibility to make sure that it's safe.
And there's a good example of this with breast pathology, doing estrogen and progesterone immunohistochemical stains. That's often sent out to a reference lab. And it used to be the reference lab would not only do the study, but they'd also do the interpretation. And that would be reported out by the laboratory of the client laboratory. Well, LabCorp and Quest and all the other large reference labs stopped doing it that way because of litigation that was occurring, not only against them, but the threat that their clients would be litigated, and they wanted to avoid that. So they actually stopped reporting out their interpretation. And then they moved to standardize the test better so that the results from the different reference laboratories would be closer to each other. So we can see that, and this occurred many years ago. This occurred, gosh, about 15 years ago. So we know there's a precedent for this. And what we're advising is that this is going to probably be stepped up, especially in the oncologic field, where you have a lot of these very sophisticated tests that are being done to determine what patients should be put into clinical trials, as well as what patients, when you have approval of FDA of a particular pharmaceutical, which patients should get it and which patients shouldn't.
Another thing that you may want to ask the reference laboratory is, how do they handle proficiency testing for these LDTs? That's another area that they have to be doing now, but you need to have confidence that they are doing it well.
So we have a question that came in. I'm not quite sure what the meaning of it. I'll just read it, Mark and Deborah. How can we regulate both private and public tests, the quality of those tests? I'm not quite sure what was meant by private and public. But generally, I think in terms of how FDA regulates things, they don't make a distinction between private and public. Any comments?
That's an interesting thought, because there are some states that have state laboratories.
Mhm.
I've seen that.
Oh,
It
yeah.
varies from state to state. I'm not sure how they're regulated or whether they only could fall under their own state regulations. What do you know, Deborah?
I don't know.
Yeah,
Um,
I would consider that to be a public,
but yeah, but they
laboratory.
have to, well, that's a good question because like New York State
Yeah,
has its own.
I don't know. I don't know
I
the
bet
answer to
if
that
the
question.
samples or the test results went outside of the state or into the state, then as soon as it crosses the state lines, then they would have federal jurisdiction.
Right, but how do that? So let's say it's a state lab. How do they acquire their CLIA accreditation? Is it a self-inspection from within the state, or is there a public health department do the inspections? I don't know. I don't know the answer to that.
Hmm.
Yeah, and if a specimen goes across state lines, it's it's still covered under the regulations.
Yeah.
The laboratory that does it, it could be anywhere, and it's still
However,
going to be
I
covered.
have inspected state labs as a service when the organization I was in was using the state lab to perform a test for us. I did do a supplier approval inspection of the laboratory. So from the point of view of a lab that might be sending a sample to the state, you still have an obligation to officially approve that as a vendor, as a supplier of services. So I've inspected state labs before for the tests that they were doing. I didn't do a comprehensive assessment of the whole lab, but I did do it in the area where the tests that they were performing. Good question.
All right, thank you. It looks like we have two more questions, which I believe we have time to answer. Let's see. The next one is how can we no, I'm sorry. Why are non-FDA approved tests not reliable and safe?
Mark?
I didn't quite get that question. What?
Why are non-FDA approved tests not considered reliable and safe?
Okay, so being able to determine reliability and safety requires that you have adequate documentation of an adequate protocol process to assure that in the laboratory. And I can tell you that what the vendors, the manufacturers go through to assure accuracy and precision isn't the same thing as what happens with a lot of these LDTs. An example of that is the use of immunohistochemical stains for the what is called PDL1, which is the program death ligand one that is used in a lot of tumors to determine which of the tyrosine kinase inhibitors are going to be used to manage it. And it's used over and over again as the tumors escape the control of each each one of these medications. They have first line, second line, third line. I'm sure there'll be fourth line pretty soon. There are three different interpretive criteria out there, which don't match entirely. And the accuracy and precision of these studies, both between laboratories, between pathologists within laboratories, and within a pathologist, if they look at it and then 6 months later they see the same slide, that's a very highly discordant. And so there's a safety issue there. In addition, just the way in which these criteria are applied, they're a ratio of positive to negative staining in terms of different types of cells. It's a mess because there's no way a human being can actually do an accurate count of all these different things on the slide. So that test is an LDT. I doubt that if it had to go through the FDA, it would be approved in its present form because there are serious risk issues to that. Now, that's just one test. There are other tests which are not, which are LDTs, and they are safe. They're simpler, more straightforward, more quantitative, and you can actually do studies to make sure that your accuracy and precision are adequate.
However, as Mark said earlier, some of these tests have been in the laboratory for many years. And if you don't have the documentation to show that they're safe, then they're not safe. You have, and so, and then not only have they been in the lab for many years, but they could have been modified from the original qualification documentation. So there's the safety issue of concordance and accuracy and precision. There's also the issue, is it thoroughly documented?
All right, thank you. For our final question, this attendee said they may have missed this, but what is the status of this law, and when would it be expected to go into effect?
We're talking about April. So we're within a month of knowing, at least initially, what's going to happen to this proposed amendment. And I think the battle lines will be formed, and I think people will be able to see, what's going on in the field. My thought is that the laboratory field itself, which is pushing back very hard, should probably be spending more time on stating a position that would be more negotiable, because really they can't defend like what I just talked about with the PD-L1 immunohistochemical stains. But I think they can defend for grandfathering in a certain class of tests. And so even though the outcome of the proposal will be within a month, it could be years before we know the final outcome of this.
Okay, that ends our Q&A portion of the presentation. I just want to hand things over to James for some final remarks before I close out the webinar.
Thanks, Jenny. Thanks to our panelists for excellent discussion, and for the attendees for the great questions and for your attendance. Again, I want to reiterate the point that understanding and being compliant in an economic way means understanding that line and walking that line. And so you have to have the experience understanding where that line is, so that you don't go too far over it and expend resources and time that you don't need to. But if you take that approach, you can implement very effective updates and remediations to these things if you need that in a cost-effective way, and it will assure that your lab stays in compliance and meets the needs of your patients. So with that, thank you very much, and we look forward to your attendance at future webinars. Reach out to us at any time if you have any questions or need any assistance. Connect to us on LinkedIn, follow us on social media. Thank you.
Thank you.
Yeah, thank you, everyone. I'll be sending out a link to the recording in a few days, and you'll also be able to find it posted on our website, labshire.com. We've got some few blogs regarding this topic, especially as things continue to change. We'll be continuing to write about it and post about it and keep you all up to date on the changes. And that concludes our presentation for today. Thank you, everyone.
Thank you.
Bye-bye.
Thank you. Bye.